Welcome, everyone. I'm JT Vannah, your host today from Neuroelectrics. We're thrilled to have you join us for this exciting panel discussion on the transformative potential of EEG as biomarkers for psychiatric treatments. Currently, psychiatric diagnoses and treatments are largely based on subjective assessments, leading to potential inconsistencies and misdiagnoses. Implementing EEG biomarkers could provide objective, quantifiable data to guide more precise and personalized psychiatric interventions. Get ready for an engaging session with our esteemed experts as we explore the future of psychiatry. This program is our second of our quarterly panel series in celebration of Neuroelectric's 10th year in Boston, focusing on themes surrounding mental health challenges. Today, our topic, EEG as biomarkers, is of critical importance as psychiatric methodology has rarely changed over the years. This summer, we'll look at brain health gets moving with Neurotech, and this fall, suicide prevention by adapting brain circuitry. We hope you'll join us for these as well. Now let's introduce our distinguished speakers for this event in the order that they will appear. First up, we have Cory Williams, who provides quantitative EEG diagnostic services, biofeedback, neurofeedback, and neuromodulation for various psychological disorders. He holds a master's in education from Western New Mexico University and a master's in psychology from Capella University. Cory transitioned from teaching neurofeedback, earning clinical certificates, and having refined his expertise under Dr. Robert Turner. Cory joined Nashville Child and Family Wellness Center in 2017, where he continues to practice, offering specialized neurofeedback and neuromodulation therapies. Next, Dr. Jeremy Slater completed his neurology residency and epilepsy fellowship at the University of Miami, later serving as an assistant professor of neurology. He worked as a general neurologist in Bismarck, North Dakota, and then focused on epilepsy and sleep medicine in Springfield, Missouri. Recruited to the University of Houston, he directed the Texas Comprehensive Epilepsy Program, significantly expanding its capacity and reputation. Dr. Slater's leadership turned the program into one of the largest epilepsy surgery centers in the country. Now with Stratus as their chief medical officer, Dr. Slater is excited about leveraging next-generation EEG monitoring to broaden its impact in epilepsy care nationwide. And finally, Dr. Aureli Soria-Frisch holds a PhD in engineering from the Technical University in Berlin. With over 20 years of experience in academia and industry, he specializes in applying AI to neuroimaging data, particularly EEG, for clinical use. He has contributed significantly to developing machine learning algorithms for neuroimaging and has published extensively in the field. Dr. Soria-Frisch currently leads projects integrating AI-driven insights into clinical practice, enhancing diagnostic accuracy and treatment outcomes. Following the presentations, we will engage in a panel discussion where our speakers will provide insights and will address questions raised. Thanks for being with us here today. This goes to both our speakers and our audience. Thanks for joining us. Now let's get started. Cory, the stage is yours.
Thanks. We're going to start with functional EEG and psychiatry. So how we use EEG in our psychiatric practice here in Nashville, and hopefully how other people can kind of get some maybe tips or pointers on how it can be useful to use in their practice. So before we even get into this, I think it's kind of helpful to think about just what is EEG in general, what does it measure, and how might it be useful? So EEG uses an electronic amplifier to measure postsynaptic potentials of pyramidal neurons. So it is a type of functional neuroimaging. It helps us to see how hard the brain is working, or in some cases, if it's not working hard enough. What we care about are what type of activity we see, and you'll see the different frequency ranges here. So what type, and then where does it occur, and then also how much of that type of activity. So this is the EEG frequency range, and it goes from slowest to fastest here. I'm going to talk about what these different frequencies mean and how we can use them to help either on the diagnosis side of things or on the treatment side of things, at least as it applies to the psychiatric practice where we work. So the slowest type of EEG activity that we see are delta waves. And delta waves are somewhat abnormal to see in a waking EEG. We don't really expect to see a lot of delta. It's much more common in sleep. If we find focal delta findings, it can be indicative of lesions or white matter damage, sometimes from strokes. If we see it in a more diffuse pattern, where we see sort of delta waves in every channel, then we can sort of infer that there may be some sort of encephalopathy that's happening, that's affecting the patient, right? Some type of disease or toxic etiology that's affecting the brain. Typically, in most cases, delta is not super responsive to medication, unless we identify the source of the encephalopathy. It's not really responsive to psychiatric medication, although there is one big exception to that, and that's when we see epileptiform discharges. Now, what we see here are very classic spike and slow wave discharges. These can be indicative of a seizure disorder, although only a neurologist can make that distinction. So we don't make that distinction in our psychiatric practice. When we see these in an EEG, we'll describe the waveforms themselves. There's a spike and slow wave discharge, but we leave it to neurology to diagnose whether or not that's a seizure. However, we do know from the literature that this type of EEG activity is more likely to respond to anti-seizure medications. In psychiatry, they are sometimes couched as mood stabilizers, so things like Lamictal or Trileptal. We don't really know to use those medications until we see this type of activity. So when you do an EEG on a client or a patient who's walking in the door without a diagnosis, this type of activity is common in ADHD, it's common in autism spectrum disorders, it's common in panic disorders, in mood disorders. Well, all of those in traditional psychiatry would kind of get assigned or prescribed different medications. But we know that this particular type of EEG is most likely to respond to some of the classic anti-seizure medication. As we move faster up the EEG spectrum, we get to theta waves. These are typically in the 4 to 7 or 4 to 8 hertz range. Theta waves are also considered a slow wave. We need theta waves. So with all of these frequency ranges, we expect to see some amount of all of them. Theta waves help with memory consolidation, emotional processing, and abstract problem solving. But when we have too much theta, that is a common marker for ADHD. In fact, 45% of ADHD cases show excess frontal or center theta. And in those cases, having too much theta is linked to typical ADHD symptoms: poor focus, executive dysfunction, trouble with emotional regulation. Here we can see some frontal theta in this EEG, here and here. And over here, we have what's called a power spectral map. So this is a way that we can process the data in a way that's maybe a little bit more visually digestible, easier to understand or easier to see. We're talking about this big red dot right here of frontal theta. And when we see this in an EEG, we know that the literature says that this type of brain is most likely to respond to a methylphenidate-based stimulant. So, ADHD can sometimes be prescribed a lot of different types of medication. And by doing an EEG, we're able to figure out which type of medication is most likely to be effective first. So it limits the number of trials we have to do, which is great for the client and great for the doctor. Now, sometimes we'll see someone with something called a slow posterior dominant rhythm. And this is where their posterior alpha rhythm actually appears in the theta range. So when you look at an EEG as it's quantified, you might see too much 6 and 7 hertz, but this isn't actually theta. This is a slow posterior dominant rhythm, and this responds to amphetamine class medications. So by doing the EEG first, we're able to kind of hone in on which type of medication is most likely to be effective. And that brings us to alpha waves, right? We just saw some slow alpha. Alpha waves are typically 8 to 12 hertz, although it varies by age. And that's the average range. So some people are a little slower, some people are a little faster when we talk about the posterior dominant rhythm. This is a good way to think of alpha, is that it's kind of the baseline processing speed for the brain. We expect to see it in the back of the brain during the eyes closed recording, but sometimes we see it in other areas. When we see it in the back of the brain with eyes closed, it's usually associated with feeling calm or relaxed. But if we see it in the front of the brain, we see it a little bit more associated with OCD, anxiety, or depression. Now, when we see frontal alpha in an EEG, the literature suggests that SSRIs may be a good fit to help with mood, anxiety, and OCD symptoms. However, it really depends on the alpha peak, and you can't know that without doing an EEG. SSRIs are a great fit for slower alpha peaks. Tricyclics and tetracyclics are a little bit better for faster alpha peaks. And if we quantify it, this is what you see in your maps. This is an eyes open alpha here in the frontal channels. Another thing that we see in the alpha range, so within that 8 to 12 hertz range, is something called mu rhythm. You can see it in C3, C4 here. Mu rhythm typically does not respond well to medication, although there is some literature that suggests it responds to oxytocin, which we would address more behaviorally than from a medication perspective. And you see it quantified in these two little dots in the midline. So this brings us to anxiolytics. We usually use this class of medication based on where someone's alpha peak is. A slow alpha peak can cause anxiety, but sometimes we want to speed that up. A fast alpha peak, which we can see here in this blue line, a fast alpha peak tends to respond best to Buspar. And this is often a driver for hypervigilance or anxiety as well. So by looking at the posterior dominant rhythm, we can see where someone's sort of baseline processing speed is. And again, that helps us to just kind of hone in on what medication is most likely to be effective. As we continue through our EEG waves here, we get to beta waves. This is a really wide range, 13 to 30 hertz. In the low beta range, 13 to 19, we typically see improved concentration. It stimulates the production of GABA. It's helpful in preventing seizures in some cases. When we see 13 to 19 hertz, we usually feel pretty good about it in someone's maps. If we see higher beta frequencies, 21 to 30, we sometimes run into anxiety, insomnia, defiance. Sometimes it can serve as a marker for inflammation. And here in this EEG, we'll see excessive beta spindling. You can see these big bursts of fast activity in frontal channels. We know that these are exacerbated by poor sleep. So sometimes if we fix someone's sleep, these beta spindles go away and their focus and their mood gets better. But sometimes we need to address it with medication, and we know that beta spindles tend to respond to guanfacine or gabapentin. And that brings us to our last EEG waveform, which are called gamma waves. These are really, really fast. They're a little bit harder to study because the frequencies are so high that they sometimes get mixed up with muscle. So it's a little bit trickier to study gamma. Healthy bursts of gamma are associated with binding networks together and with learning new information. It's associated with the eureka or aha moment. But sustained gamma, when it's bursting for too long a time, is associated with anxiety, insomnia, and in some cases, psychotic features. The antipsychotic medications typically work well if we see too much gamma. However, they do cause slowing in the EEG. So if somebody already has too much delta or theta, we may want to reconsider this one. And if somebody has any type of paroxysmal spike or discharges, we also kind of want to steer clear of these medications. So in that way, doing an EEG first can help us to make sure that the medication we're choosing is not only more likely to be successful, but safer for the client, because the last thing we would want to do would be to potentiate a seizure. Okay, so that's a quick rundown of just the very, very basics of EEG, the different frequencies and how we use those frequencies to inform our psychiatric practice. So obviously we'll get a chance to kind of talk about this in more detail at the end.
Thank you so much, Cory. That was fantastic, setting that nice platform. So now we're going to turn the stage over to Dr. Slater. Dr. Slater, whenever you're ready.
So I will issue the disclaimer that I'm not a psychiatrist. I'm married to one, but I'm not a psychiatrist. And I'm going to briefly go over just some basics. What's a biomarker? Why bother using EEG? What EEG-based biomarkers already exist? How are they used? And then some speculations on the future. And biomarkers cover a lot of territory. One definition, a measurable indicator of a biological state or function. And this covers almost everything that you can imagine. Liver function testing in the blood is a kind of biomarker. If you're thinking of EEG-based biomarkers, we have a bunch of them already. In epilepsy, spikes and sharp waves are a biomarker for epilepsy, well-established ones. In the context of psychiatry, biomarkers refer to specific patterns or features in that brain electrical activity, and you just heard about a bunch of them, that are associated with certain psychiatric conditions. So what can we use them for? Well, the classic thing is to use them to detect the presence or absence of a disease. But you can, depending on the biomarker, you can use it to track disease severity. In the area of Alzheimer's disease, predicting disease development so that you have somebody who presents with minimal cognitive impairment, which most people my age suffer at this point every time we look for our car keys, and predict within, say, a couple of years, is that going to progress to Alzheimer's disease on the basis of patterns within the EEG. But there are also developing abilities to detect therapeutic interventions, whether a drug is present or absent in the brain, whether there's a therapeutic level, whether the drug is going to have an effect, or even predict whether or not it's going to have a therapeutic effect, or whether the drug is likely producing toxicity. So once a biomarker has been developed, there are any number of potential applications that it can be used for. So why use EEG? Well, you can see a laundry list of reasons. It's non-invasive, cost-effective. The technology has been around for a long time. It's one of the only ways that we can do a direct measure of brain activity. So if all the stuff that's happening up in your brain, all the emotions that are being generated, all the psychiatric conditions, everything that's going on in a person's life is being generated in their brain, this is the one direct window that we have. It's portable. You can do very long range and repeated studies. You can use it across the lifespan. So you can use it in infants and people 90 years of age. And it has the potential for early detection of disease. So this all sounds great. And one would think by now, given how long EEG's been around, that there should be hundreds of well-established biomarkers for psychiatric disease. Well, what do we got? We have biomarkers for epilepsy in EEG. We have biomarkers for sleep spindles, slow wave activity. But in psychiatry, for an FDA-approved biomarker, there's one for ADHD. This is based on the theta to beta ratio. Even when it was approved, the FDA stated that the NEBA system couldn't be used alone. And then subsequent studies have cast doubt on the specificity of the ratio. And the clinical adoption of this specific system has been virtually non-existent. And now this is not to say that there isn't good science behind changes to the EEG related to, and you can look at either slow alpha, as was mentioned, but the specific biomarker that's in the NEBA system failed when it was applied to the larger population. And there are a bunch of reasons that these types of biomarkers fail. When you look at, though, potential biomarkers, pretty much every psychiatric condition has been assessed. Some of the publications I've gone through have looked at dementia, so you could question whether or not that's psychiatry or not. The neuropsychiatrists will include dementia because there's so many behavioral disturbances that are included in dementia patients. And you've got examples there on the right, just a handful, and I'm not going to go into the specific details of what the biomarkers are, but they give you examples of a variety of conditions where these indications for biomarkers have been published. So for autism, the Basel study, these were kids looked at from three months to 3 years of age and assessed with a bunch of clinical and behavioral metrics to determine whether or not they had autism spectrum disorder. The specificity, sensitivity were over 95% for these EEG measurements, which would make you think, well, now we've got a biomarker for autism, but we'll talk about why this isn't the case yet, but it's an indication that these changes are real, that there are changes to the EEG that occur with certain psychiatric illnesses. The Bailey paper is important because this goes beyond simply making a diagnosis, but measuring the response to TMS, transcranial magnetic stimulation, in a treatment for depression after 1 week. So they used connectivity measures and demonstrated a visible change in the patients who were responders. And this can be very important, and it demonstrates the idea not simply recording a passive EEG, but if you actually do something to either push the brain or stimulate the brain, this can be a cognitive task, it can be outside electrical stimulation, transcranial magnetic stimulation, that by pushing the brain in certain ways, you can get a much greater likelihood of pulling out something that distinguishes a disease state from non-disease state, and we can talk about that a little bit more in the discussion, or a response to a therapeutic intervention. Schizophrenia has been looked at as well. In the Lau paper, they had over 90% ability to differentiate between patients who had their first episode of schizophrenia, individuals at ultra high risk and healthy controls. And the Talakar master's thesis was a review of a variety of studies looking at multi-disorder EEG data. So this is using EEG to differentiate between bipolar disorder, schizophrenia, and a variety of others. Now, all of these, you look at this, and this is a tiny fraction of the publications that are out there. You look at this and you say, well, why don't we have these biomarkers in practice? Well, we're going to go through a series, a list of reasons why these things haven't happened yet. There are multiple studies. They suffer from methodological variability, lack of standardized protocols. So you've got inconsistent results from different research groups and settings, so that one research group will publish something, another research group will try it, but they'll be using a different set of testing parameters, and the biomarker will fail for lack of reproducibility. The studies, even the ones that I showed you, these are conducted on small homogeneous populations, and this limits generalizability of findings to broader and more diverse populations. For people who are experienced in machine learning and artificial intelligence, this is one of the biggest dangers of the types of data sets that people use. And it applies directly to clinical trials of these types of biomarkers. The other problem is EEG data is noisy and can get lots of artifacts. You need sophisticated pre-processing techniques to see the real clinically meaningful information. And if there's inadequate handling of the data quality, this can obscure the true biomarker signals. And we've seen several examples where in the epilepsy field, where you've got very promising algorithms for detection of seizures, detection of spikes, detection of other things that were developed with these beautifully clean EEG data sets. And then when you apply them to a broader clinical population, they fail because of the noise and artifact that is present in the average recording. Brain electrical activity is complicated, to put it simply. That makes it challenging to identify reliable and consistent biomarkers that are specific to particular psychiatric conditions. One of the things that gets confused all the time, or essentially pollutes the data all the time, is if you look at these publications and you say, have you assessed for the presence of sleep pathology, obstructive sleep apnea, which is as common as dandelions, and has a massive impact on all of these various EEG metrics. And for most of these publications, the answer is no. So that if you screened out the patients who had, for example, sleep pathology, a lot of times the results will disappear. Separate issue, even if you have a very accurate EEG-based biomarker, there are some ethical concerns that should be taken into consideration. Potential distress or stigma for individuals identified as at risk for certain psychiatric conditions. You can think of an early diagnosis, for example, of Huntington's from the neurology arena, And there's a lot of debate about whether you tell someone or do you do genetic testing for a predisposition for Alzheimer's disease. And there are some patients, if you ask them, will tell you, I didn't want to know. So this is something, as we develop these biomarkers, it's something that we need to take into account. The best applications often require integrating EEG-based biomarkers with other neuroimaging and genetic data, and this can be logistically and technically challenging, particularly if you're going to apply it to a large population. Now, one of the hot topics, obviously, is artificial intelligence. This is an area that I and many others have worked in for decades. Application of various types of neural networks can capture complex and nonlinear patterns in the EEG data and pick up biomarkers where visual identification would simply be impossible. Deep learning can also automate the pre-processing, feature extraction, noise reduction, and that eliminates a lot of the manual time-consuming methods that we've used in the past. This increases the efficiency. Advanced models are multimodal, so they can take in the temporal, spectral, spatial features of EEG signals. And in fact, you can integrate in some applications imaging, clinical information to produce much more accurate models. One of the big problems that people have had with artificial intelligence applications in the past is that they're not explainable. These are black boxes. But explainable AI methods are being developed so that they make the decision-making process more transparent, and we get an idea of why the model is making the determination it makes. And particularly for the advancement of the science, this becomes critically important. So to summarize, the EEG-based biomarkers show incredible promise for early detection and personalized treatment of psychiatric disorders. Key issues that remain are reproducibility, small sample sizes, standardized protocols to ensure consistent study results, taking these things from a publication from a single institution to large-scale clinical trials, which everyone in this discussion knows are expensive and a lot of times difficult to implement. The impact of deep learning, as we discussed. For the future, what we need to see are validated EEG biomarkers with large, diverse studies, robust methods, address the ethical considerations, find practical means of implementation, and then overcome all of the things that stand in the way of the psychiatrist or even the PCP on a day-to-day basis being able to use this routinely in clinical practice. And these hurdles are significant. But the goal ultimately is to be able to use these biomarkers to enhance personalized treatment. You want to be able to get to the point where you can run an EEG, not only assist in the diagnosis, and you just saw some examples in the last talk, but using the EEG-based biomarkers to specify treatments, predict response to treatment, detect likely toxicity, and create individualized and personalized medicine. And that's it for me.
Thank you so much, Dr. Slater. There's like a wealth of discussion topics right in that presentation. So thank you so much. Next, we have Dr. Soria-Frisch. Whenever you're ready, Aureli.
Okay, thank you. I would like first to thank you for the invitation to present our work on EEG for characterization of mental health conditions. And basically what we have observed is that we need a new approach for brain health. So most of mental health conditions and diseases are dominated by an assessment of symptoms to have a diagnosis, and this leads to large misdiagnosis rates. As a consequence, we have also large development costs of CNS drugs. I think, and this is a personal opinion, because the patients that are involved in these clinical trials are not well characterized from the point of view of their brain activity, and therefore, so we have these difficulties. And we think that EEG can be of help in this picture. EEG is as easy to collect data from as a wearable And it can be as informative as MRI or functional MRI, so to get functional information of brain activity. It can acquire with devices that are wireless, easy to use. You can use even dry electrodes as the one that I'm showing here in the slide, and they are certified, medically certified. And of course, you have seen some examples in the first presentation of how to get data from them. But our idea is that we can apply as well some more complex analysis, as the second presenter was mentioning, so like artificial intelligence and complex signal processing, in order to facilitate the application and the interpretation of the EEG. And with this, we think that you can conduct, for instance, patient recruitment. You can understand better the mechanisms of actions of different drugs, as the first presenter was mentioning, and you can even use it as an endpoint in a clinical trial. And I will show you now some examples on how to use this EEG and how to analyze it in order to characterize some mental health conditions. And I would like to begin with neurodegenerative diseases, and concretely with Parkinson's disease. We have conducted a study on EEG in a condition that it's called REM behavior disorder, which is a sleep disorder, and that appears 8 years before you have a conversion. Some of these patients convert into either Parkinson's disease or dementia with Lewy bodies. So we were wondering if EEG has some predictive value in this neurodegeneration. And so we have analyzed the EEG 8 years before we knew the diagnosis of these patients. And this is the way that you look at the EEG signal. So you have here the power of the signal, and then the different frequency ranges that the first presenter was mentioning. And what you can see is 8 years before these patients get the diagnosis of Parkinson's disease, which are the patients in green, or the diagnosis of dementia with Lewy bodies, so this is the group in blue. You can differentiate them very well from the group that will remain suffering only from the sleep disorder 8 years later, which is the group in red. This is another way of looking at the signal at the right part of the slide, which is looking for relationships between different electrodes and positions in the brain. And we take all these biomarkers and we use them as an input in a machine learning system to differentiate those patients that remain suffering the RBD disease, which is the red group, from those that suffer or that will suffer 8 years later from Parkinson's disease or dementia with Lewy bodies, that will be the blue group. And as you can see in this plot, you can work with the output of this machine learning system as you would work with any other biomarker. So you can find out the probability distributions, you can find out the effect sizes and the accuracy of your decision based on this biomarker, which in our case, for the distinguishing RBD from future neurodegeneration, was 91%, which means in 90% of the subjects, we were able to distinguish those that remain RBD from those that develop later Parkinson's disease or dementia with Lewy bodies. And we have recently applied this kind of slowing biomarkers, so what the first presenter was mentioning, to distinguish in Parkinson's disease patients those that suffer cognitive decline from those that do not suffer from cognitive decline. And as you can see, these are different biomarkers that measure this or characterize this slowing. So the peak alpha frequency that was mentioned before, the slow to fast ratio, the theta to alpha ratio. And as you can see, the red group, which is the group with cognitive decline, is clearly distinguishable from the group that doesn't suffer from cognitive decline, both of them being diagnosed with Parkinson's disease. You can apply this as well in the prodromal phases of other diseases, as the Alzheimer's disease. You can use it to characterize minimal cognitive impairment, as we have done. So this is using a particular biomarker that it's measuring the complexity of the signals. And yet what you can see here is the healthy controls, which is the group in red, have a larger complexity than the other groups in the prodromal phases of Alzheimer's disease, including Alzheimer's disease, minimal cognitive impairment, and even subjective cognitive decline, meaning that you can use these complexity biomarkers to distinguish subjective cognitive decline from healthy control condition in a quantitative way. So these all measures are extracted from resting state. But if you want to increase the level of reliability, what you do is basically to apply any kind of stimulation. And this is what we have been doing. So we apply, in this case, auditory stimulation in a particular protocol, which is called auditory oddball. And what you can see here is the difference between the subjective cognitive decline, which is the red group, waveform. This is the time point 0 when you apply the auditory stimulation, and then what is the response of the brain average over different trials. The blue group is the MCI with no amyloid deposition, and the green group is the group with MCI and amyloid deposition. And what you can see, there is a significant difference in the amplitude of this peak around 300 milliseconds between the group, this last group, with respect to the other ones. We have integrated all these measures in a reporting that can be used for clinical usage. So basically, we acquired EEG signals in clinical practice. We pre-process the signals for you. We extract the features and we integrate them in a reporting. We are delivering this kind of service to a neuromodulation clinic here in Barcelona. Basically, what we offer in the report is a way of facilitating reading these slowing biomarkers for assessing the clinical condition. You can use these kind of biomarkers also for validating digital health technologies. So, for instance, we have conducted a study with a company delivering glasses for photonic stimulation that are expected to have relaxation effect. Indeed, we have observed in 25 subjects a reduction in a particular marker that we are extracting related with stress levels, and this difference is significant between before and after using these glasses. This could be a more exploratory usage of EEG biomarkers, but also in this mental health domain. So we have also analyzed ADHD, so biomarkers in the ADHD condition, and we have observed what it was commented before, this increase in theta, empowering the theta band, concretely in the high theta band, that is significantly different in different areas, both in the central area. This is the one that is showing in our study the most significant results, and in the frontal area as well. We are currently conducting a support service in the development of a drug for Down syndrome. And we are working here with a complex protocol, including different resting state conditions, but also this auditory oddball condition that I was mentioning, as well as the auditory steady state response, which is a particular type of challenge of cognitive task that has the particular advantage that it shows also in preclinical in animal populations. And this is what we are observing in Down syndrome. You have observed a reduction in the activity due to stimulation in the auditory gamma band with respect to the healthy control. And this is very consistently observed in different electrodes, and you can extract a particular biomarker from this activity that shows a very significant large difference between the control group, which is the blue group, from the orange group, which is the group suffering from Down syndrome. And this is a complete portfolio of techniques, both for resting state EEG as well as for quantitative EEG and event-related responses. We include as well in these pipelines machine learning. And as I have shown, we can work in different types of diseases, including ADHD, neurodegenerative diseases, as well as Down syndrome. I would like to thank you for your attention, and I'm happy to receive any questions and to discuss in the panel. Thank you.
Thank you so much, Aureli, for that rich presentation. And thanks to all of our speakers and everyone sort of in the audience. We are just going to kick off the discussion portion. And so, thank you everyone for your time and attention. So I really, if it's okay, I want to start with something Dr. Slater, had mentioned on, because I think this is such an important point, and I'm sure, Cory, you've experienced this in your work in neurofeedback. When we have kind of inconsistency in protocols, and then there becomes this controversy with certain techniques. Like I know neurofeedback has gotten a lot of criticism for this kind of placebo effect, when it's actually the inconsistencies and the design of the studies. So if we could, maybe start with how, to go about the standardization of a protocol. So, what would be involved in making that happen? I mean, would it be like just to maybe write a publication of the importance of standardizing protocols? Because why isn't this done already? So whoever wants to interject their thoughts.
I could take a stab at it. Standardization of protocols is absolutely critical, wonderful idea, and no one wants to do it. I think of it similar to the way that I think of neurologists who are trying to differentiate between non-epileptic events and epileptic events, psychogenic non-epileptic events. And the literature will tell you that you can't tell the difference by looking at the video. And then if you ask any individual neurologist, they'll say, well, I can tell, the other guys can't figure it, but I can tell. And when it comes down to individual protocols, if you go to the person and go to their lab and say, why are you doing it this way? Part of the time they're going to give you, these are the publications, this is the rationale, these are the papers that I based it on. And if they're using a different set of papers than the lab down the street, you're going to get something entirely different. The other thing that they do is, well, that's the way that my mentor taught me to do it. And as physician scientists, we are highly resistant to the idea that anything that we were taught by our mentors is wrong. And there's actually a lot of published data that we don't practice evidence-based medicine, that we practice mentor-based medicine, and then are extremely reluctant to move beyond that. So that's part of it, is just the resistance of people to change the requirement for the standardization is in some cases onerous. You need to do this kind of testing or that kind of testing. I would argue, because I have a background in sleep as well, that everybody undergoing any type of, actually for any psychiatric disorder, should be having baseline polysomnography and sleep testing to determine whether or not they have sleep pathology. And that simply doesn't happen. Now, I'm not a lone voice in the wilderness, but there are a handful of us like that. And that's one example of where a lab could say, look, I'm trying to do this beginning study and demonstrate the efficacy of this biomarker, and sleep studies are expensive. I can't do them. And so if you try to get to those standardized protocols, there are going to be some labs that say, I'm not going to be able to do any research if I do it according to this. And that's, that's off the top of my head. I'm sure these guys.
Yeah, to kind of piggyback on that, even within databases, if you're looking at trying to create some of those power spectral maps that I showed in my presentation, different databases are constructed differently. They have different admission criteria. And so I could take one person's EEG and run it through 5 different databases, and it'll create 5 different images just from the database alone. So to get that standardized is a whole nother kind of can of worms. There's different capability within different pieces of software. So some software can map coherence, some cannot. Some software can map or can perform ICA, like independent component analysis, and some cannot. So there's a lot of hurdles for sure. And then the other thing I'd add too, while we're talking about sort of standardizing protocols, we also need to think about the differentiation between standardizing assessment protocols, and when we're looking for biomarkers, when we're looking for what does this person's brain look like, and then standardizing treatment protocols, whether that's using TDCS or TACS or neurofeedback, because in a lot of ways, those will never be standardized. Those should be tailored to the individual brain. And each brain is going to be different, even within a subpopulation, right? So if we look at 50 ADHD brains, even if they all have the same biomarker that identifies them as ADHD, they're still going to manifest that differently and still going to require different treatment modalities or training modalities.
Yeah, I think that that's a really good point. And that was, I think another thought is that, you know, because it's so, everyone's brain is different, right? So when you think about like, you know, neurology kind of focuses on the brain and psychiatry kind of focuses on the mind, like what is the mind? It's kind of maybe the subjective view of the brain, right? And so, and that is constantly changing, right? So you know, maybe like the protocol is like a sequential set of steps, and maybe in that is a manner of personalization, right? So it may be like for each individual, but there's still kind of a set of steps that everyone would be doing, and we can't expect the results to be the same, but maybe like the steps to obtain, you know, the data is the same. And then I think another potential hurdle in terms of, you know, getting, let's say, you know, a bigger sample size of just, you know, we have these small studies and we don't have large heterogeneous, you know, data. I think there is definitely a cost inhibition because you know, you know, if we want to encourage more psychiatrists to monitor the brain, to collect sleep, you know, data on their patients, you know, psychiatrists basically get paid for making diagnosis. And this also kind of brings in another portion of Dr. Slater's presentation about stigma. So, you know, whether we're getting a diagnosis from an EEG or just kind of a subjective assessment, and I mean, I say it with a grain of salt. I mean, I know psychiatrists are doing the best they can, but and I don't want to undervalue the importance of speaking to people, because I think there is no greater healing for a person than to kind of to talk, you know, their story. But I think the impression that a psychiatrist gets is subjective, right? So whether we're diagnosing by conversation or, you know, by the EEG, you know, a person can have, you know, stigma from that very diagnosis. And I think part of that is the terminology. You know, we look at schizophrenia, for instance, which means, you know, split mind, right? And that's actually not what schizophrenia is. There was a book just published called The Awakenings by people with schizophrenia in recovery. And, you know, one of them was talking about how in Japan, they've actually changed the names of diagnoses. For instance, instead of calling it schizophrenia, it's actually called integrative disorder. And so that's not stigmatizing, right? And that actually is what the diagnosis is. And so I think as an industry, you know, in addition to all these things for measurement, it's just things we can do better to have it less stigmatizing. I think it's as simple as changing the names of these disorders. Like, you know, why do we have to call it psychosis, right? I mean, nobody, like just hearing that, I think, you know, puts a chill in anyone, right? And so these are things, because we're losing too many people, right? So what can we do? And let me throw this out there, like, what can we do? And I know the speakers here are more, you know, have a neurology background, but like, what can we do to kind of to increase like the destigmatization? Like, what do we, you know, how can we do that? Just any thoughts that people have?
In the States, there was this research domain criteria. I don't know if you might be more familiar than myself, but it was a way of redefining psychiatric disorders more based on objective assessment. On it is still an interesting move. I don't know what the others think.
The psychiatrists rely on the DSM. So if you're actually going to change the names of the various syndromes, then you have to go, and I don't know what large committee they have that periodically revises the DSM, and I forget which number it's on at this point. But
5TR.
Yeah, you'd have to be in that meeting with them and saying, but I think it may be an exercise in frustration, because as soon as you apply a new name to something, the thing that caused it to be stigmatizing in the first place will eventually recur. And the best example of this is for all of the scientific names that we've developed for patients. If you're measuring their IQ scale, and then you look at the scientific names for patients who are in the lower range of the IQ scale, at some point that whatever term you come up with, doesn't matter what the word is or combination of words, it becomes pejorative. They used to use, idiot, imbecile. I mean, there were a whole variety of these words. And as soon as they use the word, then the guy standing next to the guy in the lab looks over at him and goes, "You're an idiot," or "You're an imbecile." And it becomes pejoratively applied. Even if we could change the names, I think over time, the names that we change them to would acquire that. And it requires better public education on mental illness to realize that this is mental illness is real. It's not a failure of personality or personality strength. It is not possession by demons. And that's from a public health standpoint, I think it's a terrific idea. I don't know that the will is there to do that kind of project, but it's definitely needed.
But
Yeah, and just to report, in Japan, it's been tremendously successful. So they're seeing less suicide rates. They're seeing people being more receptive to treatment. They're seeing, more people, seek out, therapy and whatnot. So, but I think, for today's discussion, I think just, having more of the objectivity, I feel like, enter the psychiatric realm, I think would be really, helpful. And I think, I think another part of the challenge too is how our clinicians are getting paid, right? So they are required to use a DSM to make a diagnosis. And, without going too deeply into this, we have, I think, Carl Rogers, like back in the 40s, like didn't see the value in a diagnosis. And I think a lot of clinicians today don't either, because I think, obviously we want to treat patients, but when we want to, people are so complex and we're trying to pigeonhole into a specific diagnosis, I think is not always precise or accurate.
Yeah, to add to that, I tell people in the clinic, when we do an EEG assessment for a new patient, I tell them I'm diagnostically agnostic. I don't really care what diagnosis you walked in with. I'm going to look at what your brain function looks like, and I'm going to tell you how that may impact your day-to-day or your cognitive function. And so whether they're diagnosed with ADHD or not, I'm going to look at different brain features and say, hey, you've got X, Y, or Z, and this is associated with scattered attention. Now, whether that fits an ADHD diagnosis or not, I don't really care. Your attention probably suffers because of this, and that's what we're going to work on when we do our clinical work.
yeah, I completely agree with this. It's a change in the paradigm in the way you are using EEG to understand what is functionally not working in the brain. And basically this can be associated to different diseases. And this is also what we find, that there is a lot of comorbidities among psychiatric diseases, and might be also because the mechanism, it's wrongly functioning is the same. And sometimes you are diagnosed as something, and another moment you are diagnosed as another thing, because basically the focus was put in somewhere else and not on what was functionally not working in the brain. And if you are denoting what is functionally not working, I think we would do a step out of this stigmatization, because you will say DSM whatever network is not right working, or whatever, we need to put it back in shape, or your activity is unbalanced in your brain, or whatever. No, that's another thing.
This is where cognitive testing paradigms, I think, become important. One of the problems, for example, if you're trying to create an ADHD biomarker, is, okay, give me a population of patients with ADHD who have no comorbidities. And that's really hard to find. And so the baseline data is messy, but you're absolutely right. Particularly if you're doing either electrical or magnetic stimulation, which can be, objectively and consistently repeated, or doing cognitive testing, oddball paradigm, and there are many, many others that have standardized protocols. They are stimulating the brain in one particular fashion. They take advantage, all evoked potentials take advantage of the fact that the brain is presumably going to respond the same way to the individual stimulation each time. And then you average it together and you're looking at just that brain response. All of those things can give you, regardless of the diagnosis, like you said, they give you a series of objective metrics. And putting those together, I don't think it's been, or maybe you guys are doing it, but I don't think it's been done consistently enough where people can say, look, we have these diagnostic groupings. This group of people who respond to this medication have these particular abnormalities on this series, on this battery of tests. And traditionally, they were all called schizophrenic, or traditionally, they were all called X, Y, or Z. Probably better integrating it with the existing clinical diagnostic criteria, because if you don't, it'll never get accepted. But there is a pathway there, I think, that you could move forward with. And again, it would come down to standardization. So you'd have a bunch of different labs and different populations all using exactly the same battery of tests. How you convince the group of us to do that, I have no idea. But it's not a bad thought.
Yeah, and also just speaking on, you had mentioned medications. Today it's so difficult. It's pretty much trial and error. And there's a pharmaceutical company that kind of recognized this, and they said, well, because it's an error in the trial, right? So instead of, could this be a better path to improve treatments? So when we look at the amount of money going into pharmaceutical development, and such a small percentage actually makes it to a marketable product, right? And so, right now, the animal model is still the most common way of clinical trial development, but could mapping the brain through clinical trials, give us another set of tools to have more precise medications to look at, to be able to track not only the response to the medications, but the side effects. It's heart-wrenching to see so many people suffer. And, unfortunately for the psychiatrists as well, it's like, well, it becomes trial and error. Like, this medication didn't work, let's try this one. And I don't know if people have thoughts on kind of, using EEG for clinical trials to develop better, pharmaceutical products, and even we've looked at, electrical stimulation devices, both TDCS and TMS, that for refractory patients that are not responsive to medications, that with stimulation, they become responsive to treatments. So that's sort of combining two different questions, one with the EEG for to testing like the medications, right, to get a more precise product, and also for patients that have not been responsive to treatments. If we add, and we didn't talk that much about stimulation today, Aureli did a little bit, but using that to have people become, responsive to treatment. So I find, neurotechnology offers a partnership with, pharmaceutical companies. I think so much, people see it as one or the other. Like, either I'm going to do electrical stimulation or, TMS or pharmaceuticals, but like, what if we, rather than either or, like this emergence of a partnership to help the patients like become responsive. So I don't know if people have thoughts on that.
I have good news for you.
Yeah.
The one is that the pharmaceutical companies are increasingly recognizing the need to do concurrent EEG recordings. And this is something that we do at Stratus. And many other companies are doing this as well. But they're increasingly interested in getting baseline and post-treatment EEG recordings looking for biomarkers of efficacy, looking for biomarkers of toxicity, the presence of drug, all of the stuff that I mentioned before. And more companies are beginning to do this. The other good piece of good news is I know that from in the psychiatric community, and Cory can probably comment on this as well, but that there is a rising number of physicians who are doing both, who are doing TMS plus drug, or TDCS plus drug, et cetera. And you're right. I think the combination of it is one more therapeutic approach. The epilepsy parallel is people with vagal nerve stimulators. Well, we had an implanted device for control of epilepsy. We never took them off their medicine. I had in 30 years, I had one patient who was on VNS alone, and we implanted thousands of patients. They were all on VNS plus anticonvulsant, and the two things were working synergistically. The beauty about electrical stimulation is that it doesn't have drug side effects and doesn't have drug interactions. So that you can get that kind of potentiated effect.
Yes, excellent point.
Yeah, and the research on TDCS and TMS both indicates that it increases neuroplasticity, right? And so if we as a psychiatrist, if that person is thinking, I think this medication is going to be effective for this patient, and then through stimulation we improve neuroplasticity, we would expect that medication to be that much more effective, right? If the neurons are more open to rewiring and to changing the way that they interact with each other as a result of stimulation, then the medication, at least in some cases, in theory, should be even more effective. So I think a combined approach is absolutely the right one.
And coming back to the usage of EEG, I think so. It is clear that the costs in CNS drug development are too high, are the largest one in all pharmaceutical domains. And therefore, the pharmaceutical companies need to do something different. So it can be the combination with transcranial stimulation. It can be as well the usage of EEG to have more homogeneous populations from the electrophysiological point of view, in which you would expect the drugs to have also a more homogeneous effect, and therefore you can show some efficacy. That would be also my approach, and also this of the company that you were mentioning before.
Yes, thank you so much. I know that we could probably spend a few more hours. I mean, there's so much to discuss here, but let's say if each of you kind of wants to share a thought on what we can do, us individually, and I think audience members can do to encourage more objective data being used in psychiatry. What is something that each of us can do, whether it's reaching out to, I feel like there's a legislative element here too, just because of insurance billing, because I think the other question is, I know, in neurofeedback that even states that do kind of approve the insurance, I think it's some states it's for, $20, and it should be, at least, 150 or more. So just with a thought of what we can do, what everyone could, because, it's nice to talk about things and have discussions, but things are not going to change without action. So what are some actions that each of us can do to promote, more work? Let's get, these more data on, using EEG as biomarkers in psychiatry, right? So what are, because we have just a modicum of publications, like what can we do to have, repeatable data that we can use and share? So I'll leave that for each person, whoever wants to start, with your final thoughts on kind of, moving forward and putting these thoughts into action. What are things that we can actually do?
Yeah, so I'll jump in. One of the people that I had the privilege of learning from was Jay Gunkelman, who's a very famous sort of EEG analyst and well respected in the neurofeedback field. And one of the things he told me very early on was, you have an ethical obligation to publish your data if you do something that you haven't seen in research before. And I think that we have a lot of really smart people in our field who are mixing modalities in really brilliant ways and who are helping patients get better all the time. And none of that data becomes publicly available or published. And even if it's just a case study of, I saw these 3 people, they presented similarly, their EEGs looked similarly, and this is what we did for them. Publish. And eventually you can kind of get the ball rolling on, other smart people, they read those papers, they start thinking, and we get bigger and bigger studies and more validation. So
That's a great suggestion. Thank you so much. I feel like not enough clinicians do publish. Thank you so much.
Boy, it's a tough question.
Yeah, sorry, I know it's
I won't disagree with the idea that particularly the physicians in private practice, I can think of multiple instances. The use of topiramate for migraine, for example, came out of private practice observations. Many of the applications of anticonvulsants in psychiatry, a lot of those initially came out of private practice applications. As a neurologist, we always say that the psychiatrists steal our drugs as soon as they get released. But smoothing that path and creating, and I guess you'd have to pick a disease state and demonstrate, because right now, as I pointed out, we have one FDA approved biomarker for a psychiatric disease.
right.
There's nothing FDA approved for schizophrenia or bipolar disorder or any of these other things. And it would take the collective will of the community to come out and go, here we have solid, and there's, as you saw, there are lots. I gave you 4 publications out of thousands. There are lots of publications in each disease state where you could put a group of researchers together and say, this is the best possible protocol from our existing evidence. Let's push that through and use the same thing in all of our different labs, all of our different clinics, and overcome all of the things that prevent it in terms of different equipment and different protocols and all of the things that contaminate the data, to get to the point where you actually have something that is going to work for the average clinician and get that FDA approved, because that first hurdle hasn't been passed yet. We have yet to produce a biomarker that's FDA approved, clinically used all the time, where people can say, oh yeah, EEG-based biomarker, well, that one we use every day. Once you see one, then it becomes easier to go, okay, well, we did it for schizophrenia, now we're going to do it for bipolar disorder. Now we're going to do it for depression. And from the reimbursement question, I don't have a good answer for you, because I know, for example, when vagal nerve stimulation was studied for depression, the data for depression is better than the data for epilepsy, solidly better than the data for epilepsy. It got FDA approval and then went through CMS and the RUC, and they looked at it and they said there are 20 times as many patients with refractory depression as epilepsy patients. This is going to cost us a fortune. So we can't afford it. And then what they picked apart was the definition of refractory depression and didn't approve paying for it. And I don't know what the answer is to that, except for maybe coming up with, cheaper interventions. At least EEG is relatively inexpensive.
Right. Okay, thank you so much for your thoughts.
What I think what we can do is basically what we are doing, which is to facilitate the access to the EEG, because it has a lot of variability. And this is what we are trying to de-risk a bit the usage of EEG with expert knowledge on it. This especially deals with, or it's related to the usage of evoke-related potentials, which I think are a good way to go to increase the reliability of EEG as a tool, as a diagnosis tool, but also as a stratification tool. This would boost the usage of these evoke responses and not working so much with resting state, which is much more, it's much easier to acquire, but it's much variable from subject to subject. And I think also one interesting point to work on is on the existence of normative data sets that are available for everybody to work with. And this would facilitate as well to understand when an EEG signal is going or splitting from normality, and what this could be, and trying to interpret why this is happening.
Yeah, that's a great insights. And I think that goes back to what Cory was saying. There are like these databases that exist, right? And that they're not consistent though. So you said you could plug in data in one. So it goes back to what Dr. Slater was saying about the standardization, right? And so I guess these are things that we can, I guess, collectively work on. You know, first is just even being aware that we have these discrepancies, right? And so what can we do to normalize them, to make, so thank you everyone for being here today with your presentations and thoughts, and thank you everyone in the audience for joining us. And I hope you will come back this summer for Brain Health Gets Moving with Neurotech, and continue, this. I think this issue is so important, and I think this could bring, such, greater hope to the psychiatric community, both for practitioners and more importantly for the patients. And thank you here today for your efforts in this cause. We appreciate it.
Thank you.
Thank you.
Take care.
Yeah, thanks.